Hello, and welcome to today's presentation, and thank you for joining us. I'm Noah Koenig, Marketing Director for PrecisionLife, and your host for today's webinar. And I'm delighted to say that I'm joined by Steve Gardner, a pioneer in precision medicine and data science and CEO of PrecisionLife. Great to have you with us, Steve.
Thank you.
I'm really looking forward to the presentation. The studies and findings that you're going to be sharing are incredible, and the potential implications for healthcare, nothing short of remarkable. So I've no doubt that those of you who are joining us will get plenty out of this. And just a quick bit of housekeeping before we get into proceedings. If anyone has any questions at all about any of the information that's being presented, or would like to know more about any of the programs or research that Steve will be sharing, please do send us a message in the chat window, and we'll do our best to answer those questions either during the session towards the end, or by contacting you afterwards. Okay, so let's get started. Steve, over to you.
Thank you, Noah. I'm delighted to welcome you to this webinar. As Noah says, I've been working in the precision medicine space for over 30 years. And in that time, we've seen the field progress enormously. The Human Genome Project and its consequential developments have transformed the care of oncology populations and rare disease populations, and turned diseases that were previously defined by the anatomy in which they occurred into things that have a molecular definition. The challenge that we've got is that those same benefits haven't worked their way through into complex chronic diseases. And this is a real challenge for the affordability of healthcare, the delivery of solutions that actually help billions of patients around the world. And as the world gets older, as the populations increase and become more morbid, we're ending up in a situation where it really is becoming a struggle to manage disease. So turning that question on its head is really the subject of this webinar, and thinking about how we can use precision medicine to increase health span and prevent complex chronic diseases. The easiest way to afford healthcare is for people not to get sick. And while that's very easy to say, I'm going to talk today about some innovative approaches, tools, technologies that can help us understand the drivers of those disease biology, help us predict the risk of disease in patients, and find solutions that are going to help prevent the onset and progression of many of these more complex chronic diseases. So this is an overview of the talk. I'm going to start just by setting the scene in terms of the healthcare costs and challenges that are faced by the sector, particularly in the context of complex chronic disease, and talk about some of the things that we need to do. So particularly looking at getting better and earlier, more accurate diagnosis, and understanding the specific disease drivers that are working in individual patients, such that we can then understand their risk and find solutions that will prevent the onset of disease. And finally, I'm going to talk about a really exciting technology, which we're calling active protective biology, which is really turning the problem on its head and looking at those systems in the body, those cellular processes in the body that are working to resist disease pressure. And we're starting to be able to find those in a really scalable manner. And I think this is one of the most exciting new opportunities. So let's start with where we are with healthcare. Healthcare consumes a huge amount of resource, about 18% of GDP in the US and about 12% in the UK. The challenge is that consistently over the last 60, 70 years, the proportion of GDP spent on healthcare has been growing faster than that GDP rate by 2, possibly even 3% per annum. And this obviously means it's becoming proportionally more expensive, and healthcare, in a very real sense, is becoming unaffordable. A lot of that is because many of the population have not even just one, but in fact many chronic diseases. And some of these are incredibly expensive. So, US healthcare spending, 15% of that goes on diabetes treatment. So, that in effect is about 2% of US GDP just spent treating diabetes, which is a huge burden on society and on the patients and the carers for those patients. Here's the real challenge. As our society ages, there is a point around about 65 years old where the number of chronic conditions that a patient suffers from starts to increase, and that puts a massive burden on the healthcare system, which is now having to deliver more care, more interventions, more drugs to those patients, largely because it's treating sick people. So it's treating people as they become sick and as they present with their symptoms. The difficulty in most developed economies is that that proportion of over 65s is going to double in the next 20 or 30 years. And this is a real challenge for planning on how we're going to deliver healthcare. The reality is we need to intervene before we get to presentation with dementia and diabetes. And so the question is, how can we do that more effectively? One of the benefits of uh, the Human Genome Project and precision medicine in the context of oncology is that, as I said, it became easy to find genes that were involved in disease, and therefore we targeted diagnostics against those genes, and we targeted therapeutics against those genes. Um, and those genes drive the disease in a very real sense. If you have a mutation in, for example, BRCA, it leads to a compromise of your genome surveillance apparatus, which leaves the body open as oncogenic changes happen to the formation of tumors. Um, Unfortunately, the disease biology of complex diseases, as the name suggests, is not that simple. And the tools and techniques that we've developed don't work as well in complex disease biology. And this is a really good example. This is a genome-wide association study in early onset Alzheimer's in a UK biobank population. And you can see that we're not finding very much information. Each of those dots represents a mutation or a SNP. Um, and all of those are in a single gene. They're in the classic gene ApoE4. So looking at this, you would expect that that's the only drug target of any note in this disease because you can't see anything else. Unfortunately, we've had about 130 failed clinical trials in Alzheimer's targeting largely this lipoprotein ApoE signature. And in many ways, that's a little bit of a failure, not so much of our ability to produce effective drugs, but to find the right patients for whom that is actually the mechanism driving their disease. And so we wanted to reimagine the way in which we analyze disease data. And we started to identify combinations of features that were associated with particular subsets of patients that allow us to understand what are the specific disease drivers that are creating this disease pressure in those patients. And so you get the graph on the left from exactly that same data set. And what you can see now is a whole series of stratified patient populations. So the colors represent different subgroups of patients who have a different mechanism, different gene or target causing their disease. And because we can relate these back to individual patients, as opposed to being a population average, we can now start to evaluate the prevalence of those mechanisms within this patient population. So it turns out that for this blue group, which are the ones who would benefit from an anti-amyloid, anti-tau lipoprotein drug, um, they represent about a third of patients, which explains why it's been so difficult to put effective medicines into the market. But there are other mechanisms that we can go after. Obviously, understanding who is going to benefit from any particular drug therapy is absolutely essential if you have a disease that's as heterogeneous as Alzheimer's here. Most of the complex chronic diseases that account for about 80% of healthcare spending and which affect billions of patients around the world are as complex as this. They're as heterogeneous as this, with multiple mechanisms driving disease. So understanding that, being able to identify all of those features that are driving disease is absolutely essential in being able to predict and prevent disease. But it's not just genomic information. We also rely upon large-scale electronic healthcare records and longitudinal clinical histories, epigenetics, proteomics, transcriptomics, and a wide range of other markers. Fortunately, there are now a whole series of data sources, some large-scale national data banks, some healthcare systems, research consortia, patient charities, who have been collecting these diseases, data on these diseases at a scale that is suitable for their analysis. So I'm going to show you a few examples of this in the context of solving some of the problems that we talked about earlier. But bear in mind, through this, the critical feature of a precision medicine approach is that we can not only identify the drivers of complex disease biology, but we can also find the patients for whom that mechanism is the one that's driving their disease. And we can also reduce these to practice, as I'll show you, in terms of a new class of tests. We're calling these mechanistic tests. So tests that reveal the underlying disease mechanisms driving illness within specific patients. I'm going to start off in endometriosis and just talk about the need for better diagnostics inside patient populations. Endometriosis is a massively underserved disease. It affects about 1 in 10 women during their childbearing years. It's about 190 million patients worldwide. And those patients have debilitating pain, for many of them for weeks in a month. They suffer because of this and the delay in getting a diagnosis. They suffer depression and anxiety, uh, as I said, deep pain. They have higher rates of mental health disorders, ovarian cancer, infertility, and it affects their ability to participate in the workplace and in society. On average, though, it still takes about 8 to 10 years to diagnose the disease. And even then, that diagnosis is only confirmed by surgery. So we have a very expensive disorder that is debilitating to a huge number of patients, and an ability to actually go in and, if we solve this problem, improve the health of those patients at the same time as reducing a series of costs to the healthcare system. And that comes from increased visits to primary and even secondary care through OBGYN, through specialist reduction in specialist referrals, and a reduction in unnecessary surgery. And these costs mount up. These are expensive uh, interactions with the health system, which is all represents clinical waste because it is not actually helping the patient. So being able to do something about that, at the very least, being able to reduce unnecessary surgery would be a hugely beneficial thing to do. So one of the things that we're looking at is being able to identify risk signatures associated with specific diseases. And we can use these for differential triage. In other words, we can differentiate between multiple disorders that appear to have the same type of symptoms, in this case, deep pelvic pain. And we can, by building a profile of the drivers of disease across a broad range of indications, and then testing a patient to see which of those signatures they have and which disease they're therefore most likely to have, we can give a good indication that confirms whether this patient should indeed go for laparoscopy and confirmatory surgery. Indeed, are they likely to have endometriosis, or is there another explanation for their symptoms? So I'm going to talk a little bit about the type of tests that we're building here and what they can be used for. We've called these mechanistic tests, and that reflects the fact that these are not only identifying disease risk, but also picking out the causative mechanisms underpinning those diseases. And this is a little bit different to what a traditional diagnostic tool would do. Traditional diagnostic tool is finding a metabolomic biomarker that usually increases when a disease is present inside a patient. So they're not really telling you what's driving that disease, but they are giving you perhaps the stage of disease, maybe if you measure it multiple times, the rate of progression. But what it isn't telling you, as I say, is what is the risk for that patient or that person before they start to show with symptoms. And critically, what are the mechanisms underpinning disease? Because those mechanisms are going to tell you which drugs are most effective for those patients. So having a direct connection to the mechanism of action, the likely effective therapy, the differential triage, all of these are incredibly clinically actionable, and they work across diseases that don't have traditionally good diagnostics. So you can think about the endometriosis case here. I'm going to take us into a slightly different case study that has similar challenges. And this is a disease called myalgic encephalomyelitis, or chronic fatigue syndrome. And this is a disease that is massively underserved. In fact, uh, prior to our recent study, there was no biological basis for the disease understood, and therefore many clinicians um, to this day refuse to believe that it's actually a real disease. And yet, many patients suffer from debilitating fatigue, cognitive impairments, uh, chronic pain, and a whole variety of other debilitating symptoms. When we started to analyze the the disease, there were no genetic associations that had ever been identified. We were able to identify a series of genetic associations, so SNPs, that were associated with the disease. And we've now replicated these in 3 different data sets. So that's great. That's a good starting point. But of course, what you want to do is make sure that these are associated through a mechanism, and that that mechanism is connected to the way that those patients present in the clinic. So we clustered um, the mutations and the disease signatures by the patients in which they co-occur. And that's really what you're looking at here. Again, the dots represent individual mutations or SNPs, and the connections mean that they co-occur in more than 20% of patients. And this allows us to cluster up by the patients and then see whether those patients present in a consistent manner. So here for this green group, the main gene involved here is a gene called ACAP1. And this is involved in the replenishment of cellular energy supplies after exercise. It's a little bit like recharging a battery. If that mechanism is broken, you would expect that these would be the patients who have post-exertional malaise, that deep energy crash. And that's exactly what the green patients who have this signature present with. They're highly enriched for fatigue and post-exertional malaise. Versus the group next to them who have defects in neurotransmitter biology, and they are unable to produce GABAergic neurotransmitters. And you'd therefore expect that these would be the patients who'd present with cognitive impairment, mood disorders, and if you analyze their blood, you'd see higher levels of neurotransmitter precursors. And that's exactly what these blue group of patients present with. And there are many other examples. So this is the clock gene involved in circadian rhythm. This is the insulin receptor involved again in metabolic control. And all of these are consistent with the way in which the patients who have these signatures present. So we've teased apart a disease where we had no genetic associations, we didn't know which organs in the body were involved, there were no diagnostics and no effective therapies, into something where we can now start to apply precision medicine approaches, not only to better diagnosing the disease, but also doing something about treating the disease. So we started to look at this in the context of diagnosing the disease. And we took a very simple view, which was that we should count the number of times we saw the combinations appearing in patients versus controls. And the two distributions are quite different. So you'll see that there are many cases that have many of these risk signatures. And there are many fewer healthy controls that have lots of these risk signatures. That difference is equivalent to the predictive value. And the predictive value here is actually it's surprisingly high for a disease that didn't have previous genetic associations. So what we've been able to do is create a predictive risk score that we can actually reduce very easily to a practical test that can be used in the clinic or indeed at home. And we can do that because there are only 200 SNPs here. And it's very easy to test for those on a genotyping array. A genotyping array is a specialized type of chip that can detect particular pieces of DNA that represent certain sequences in which mutations are most likely to occur. And this is the type of technology that was used, for example, to characterize all of the participants in the UK Biobank. So 500,000 patients. It's a very cost-effective and pretty accurate way of identifying genetic features that can be associated with risk, disease risk. And we've used this so we can now take a saliva or a blood sample from a patient in the clinic, or they can even do this at home. It's a little bit like doing a COVID PCR test. And we can go through and evaluate which of the disease signatures we can see. We can also go and evaluate which protective signatures we see. And I'll come back to this, but there are processes in the body that work to resist disease, and understanding how many of each of these a patient has gives an overall disease risk score. And that's what we can use for differential triage. But of course, what it's also doing is giving us a mechanism by mechanism score. And that's great because that then says what is driving their disease, which symptoms do we expect that patient to be presenting with, and which therapies are most likely to be effective for them. So we have a clinical decision support tool that synthesizes all of this information and turns it into a clinical report. And that clinical report is a very detailed document. It mimics in many ways the type of report that you would see in precision oncology, where you get the specific clinical variants that are driving various cancers, and it will tell you which therapies are most likely to be effective for that patient. So we're mimicking that, but in a much more complex way because we don't have one mechanism driving disease. We have seven in the case of ME/CFS. So here you can see for patients 3 and 4, their disease is primarily a metabolic and inflammatory or autoimmune condition, and they look very similar to each other. Whereas patients 1 and 2 have much more impact around that neurotransmitter dysregulation. So understanding this helps characterize those patients and give identify ways of treating them that are going to be suitable for them, even in the context of a complex disease. So the mechanistic tests are a really important part of this story. They allow us to stratify patients by mechanism and show their most likely effective therapies. It's a very simple, cheap, non-invasive test. It has the potential to scale across many different diseases. And it's a very flexible platform. So it can be used not just in the healthcare settings that I've described, looking at disease risk, allowing prediction and prevention of disease and selection of personalized therapies, but also even in biopharma. So used as a test to select patients who are likely to respond to a medicine, you can then use it as an inclusion criteria to design smaller, more targeted, faster to read out, more likely to succeed clinical trials, and also as companion diagnostics, um, as a tool to identify patients as you go into the clinic with a new medicine who are likely to benefit from your therapy. We have this level of detail necessary to run this across about 50 diseases so far. And in fact, we have data to support another 75 diseases. When we look across these, they represent about over 5 billion lives or patient lives. Of course, many patients have multiple diseases as well. But these are very poorly served by precision medicine and genomic medicine approaches, in contrast to things like oncology and rare disease, which are much better served. So being able to extend that capability into these disease areas can have a profound impact on patients. And I'm going to give you an example in the context of type 2 diabetes. So I mentioned earlier, the overall statistics of healthcare spending. Um, Type 2 diabetes, just in the US, accounts for roughly $450 billion a year. There are around about 470 million diabetes sufferers globally. Um, Oddly, only 20% of those are over 65. And nonetheless, it's approaching $2 trillion per year spent on treating type 2 diabetes. Now, that isn't spent on metformin and insulin. 80% of that is spent on treating disease complications. And this is where patients cannot manage their disease and they progress into strokes, into dementia, into neuropathy and chronic pain. They get retinopathy and blindness, they get ulcers and amputation, and they get renal failure, so kidney failure, that leads to them needing dialysis or transplant. And these are the states that become incredibly debilitating, life-threatening, and expensive to treat. So we're looking at ways of identifying early, more accurate diagnostics that can evaluate a patient's disease risk and provide a window into which we can deploy different interventions to change the progression, to change the trajectory of disease inside a specific patient. So we analyzed from UK Biobank a population of type 2 diabetes patients who had recently been diagnosed and progressed to these complications very quickly. And we compared them against patients who'd had the disease for decades and tolerated it without complication. And we looked at the differences there to separate out the factors that were driving risk of renal failure, acute presentations with ketoacidosis, cardiovascular risk, neuropathy, and ophthalmic effects. And we have a risk model that can predict this just from the genomics with some social determinant of health information. And because that can be used at any point in the disease process, it can be used right at the earliest diagnosis of type 2 diabetes or even pre-diabetes to say this is a patient who's most at risk of renal failure, for example. Now, we can do something about that. We can test for blood pressure. We can obviously do continuous glucose monitoring. Um, we can change their lifestyle, so we can get them to reduce salt intake and blood pressure. We can change how obese that patient population is, perhaps with GLP-1s. Or we can also target a specific therapy. So we were able to identify not only the patient population most at risk of renal failure in type 2 diabetes, but also those that would respond to one of the existing drugs. This is a mineral corticoid receptor antagonist, a drug called Kerendia, which is now approved in the EU and US for the reduction of chronic kidney disease progression in type 2 diabetes patients. So being able to know that and being able to prescribe this drug earlier in a targeted manner will bend the progression curve away and prevent those patients potentially from even experiencing any symptoms of renal failure, let alone needing dialysis or renal transplant. So that's clearly an incredibly important thing to be able to do. But there are some other things that we can do, and this gets really exciting. So this is thinking about the challenge of disease biology in a slightly different way, and looking at those things that the body does to resist the progression into chronic disease. And I'm going to take you through a case study in a neurodegenerative disease, ALS, also called motor neurone disease in the UK. And we've spent a lot of time looking at disease risk in terms of genes that are associated with causing the disease. But we've also applied a lot of the same principles that I was just describing to start to think about, are there mechanisms, a little bit like BRCA1 and 2, prevent copying errors within the DNA of an individual manifesting as new tumors? Are there processes that we can identify that are acting to resist the disease pressures that potential ALS patients find? So we can do this by going back to that high risk cohort of the healthy controls, so people who don't have the disease. Um, what am I talking about? We've got a whole population of patients, as I showed you in the ME/CFS example, where some of them, right at the top end of the healthy control distribution, have many, many genetic risk factors. And we want to find those that have also had exposure triggers from an epidemiological or environmental perspective, and who also are as old as possible. So they've had as much time to get the disease, and yet they still don't get the disease or anything that looks like it. And I'm going to call those our protected cohort. So these is a really interesting set of patients. The question obviously is, why are they not getting the disease? And so what we can do is take that patient cohort, compare them to patients, and identify the things that make those protected people different from ALS patients. And that gives us a set of targets that we believe are enriched for these actively protective disease processes. And this is a fascinating new set of targets. These we can use to create a new generation of prophylactic drugs, things that work to augment processes that are protecting the body against disease. And of course, as we move into a world where we have therapeutic mRNA vaccines, those are really turning your muscle cells into little factories to do precision protein production. And of course, instead of, for example, producing SARS-CoV-2 spike proteins, we might use them to produce these protective proteins. And that augments this natural protection. So we ran this study in ALS, and we found 21 actively protective genes. And we presented this work at the NEALS conference in November. And we talked about three of the genes that we had identified there. The first was an activator of a gene which actually is an oncogene, ERK1 and 2. It's long been associated in the literature with neuroprotection in the context of ALS. Because if you inactivate this, the ERK1 and 2 pathway, you get a neuropathology, you get neurotoxicity based on glutamate excitotoxicity. So finding that was a very positive and reinforcing kind of finding. But we also found two other genes that were particularly interesting. So the first was GRIP1, which is glutamate receptor interacting protein. And this had been reported by the group at Duke under Rick Bedlack as being associated with those extremely rare cases where ALS patients go into remission. But they'd not been able to explain the mechanism or get any confirmatory evidence for this finding. Then the third one was a gene called MTRR, which is involved in methylcobalamin or vitamin B12 metabolism. And this is fascinating. When we go in and look at this pathway, from the analyses that we've done, we actually found a whole range of different associations. These genes, cubulin, megalin, and the transcobalamin receptor, all show up in our risk analysis, because if you break them, it prevents vitamin B12 being transported or being absorbed across the gut, moving into the bloodstream, and then being transported through the lysozyme and fulfilling its main functions. So preventing that is obviously a bad thing. We've also found this in our protective analysis. So what we think is going on is that this gene, this MTRR gene, is responsible for turning homocysteine into methionine, and homocysteine is a neurotoxic metabolite that appears at very high concentrations in the cerebrospinal fluid of ALS patients. By augmenting this mechanism, you increase the transformation of homocysteine into methionine, and it reduces that neurotoxic burden. And so this could well be a protective effect that will work across all ALS patients. So unlike a drug that targets one mechanism that may only work in a few patients, the protective mechanisms are likely to support everyone who is giving them. We didn't know this at the time, but actually the high dose vitamin B12, a product from Eisai, has been approved for slowing the functional decline in ALS patients in Japan. So again, a very interesting, supportive piece of evidence around this finding. But of course, we also have the biomarkers that can support patients who would particularly benefit from um, impacting this vitamin B12 pathway. So this is I think indicative of a new direction. We can do this at scale. We can do this across multiple diseases. In fact, if we go back to ME/CFS, two independent runs of this analysis identified the same gene here associated with the catabolism of tyrosine. Tyrosine is a precursor of dopamine, and dopaminergic biology is really important in ME/CFS. So being able to provide even something as simple as a tyrosine supplement is likely to benefit those patients and reduce the burden of their symptoms. Of course, I've given you two disease studies. We can do this across all 50 diseases, and the fact that these affect billions of patients, I think, gives us new tools and new techniques, not just for treating people when they're sick, but from identifying patients who have high risk of disease, intervening with them, and also finding ways of augmenting their internal cellular and biological processes that are working to keep them healthy. And if we can do that, we can move more patients away from the most extreme presentations of their disease. That allows us, I think, to reduce the burden of disease, not just in those patients, but across the health systems that are treating them. And so bringing it all together, accurate diagnosis is really important. It allows you to predict disease risk. It allows you to understand the likely prognosis and trajectory of disease within a patient. It allows you to select the optimal therapies for them. Then we can come in and start to evaluate some new opportunities, both in terms of perhaps precision medicines, either novel or repurposed, and actively protective drugs. Then we can go in and monitor those patients, see how they're benefiting, and all of that together will reduce the burden of disease, the cost of treating those diseases, reduce the drug burden on patients and the side effect profiles that they have. It'll prevent visits both to primary and secondary care, reducing the clinical wastage. And through all of this, it should help many, many patients stay healthier for longer, increasing their health span. And that's a fundamentally critical part of delivering an affordable and effective healthcare system in the future. Thank you.
Thank you, Steve. Brilliant presentation. And thank you also to our audience for sending in your questions. I've been trying to keep up as we have been going. So, Steve, if you're happy to, let's see if we can answer a few of those right now.
Sure.
Okay, so let's have a look. First question. Could you elaborate on how multi-omic data integration enhances the discovery of novel therapeutic targets for age-related diseases within your platform?
Yes, I mean, I think as you've seen, you can get a lot more signal out of just the genomic data than perhaps has been possible using existing genomic analysis technologies. But of course, genomics is not the only thing that is driving disease, and it's not the only thing that can confirm the relevance of a particular target or mechanism inside a disease. So we're fascinated by proteomics in terms of being able to look at different splice variants for a given protein product of a gene. We're fascinated by expression within single cells that are representative of the tissues involved in disease, and particularly with epigenetic signaling or methylomic profiling, where we can look at the relative amplification or inhibition of the expression of particular proteins or combinations of proteins that we think are associated with those drugs. And all of that evidence together is much more convincing and compelling when you're starting to look at those novel disease processes.
Thank you. Next question. How scalable is this approach for analyzing large-scale genomic data from diverse populations, and how does this impact the generalizability of the results?
Oh, it's incredibly scalable. I mean, we can do up to hundreds of thousands of patients in whole genome data. So scalability is not really any more an issue. You raise an interesting question though on the generalizability of results. So I didn't specifically talk about this, but for example, in endometriosis, we have some very good examples where there are big meta-GWAS studies out there that have collected data on 60,000 patients and 700,000 controls. And they published a paper in Nature Genetics where they used that using a GWAS to identify 42 SNPs. Now, unfortunately, those SNPs didn't translate very well across different patient populations. And this is one of the known challenges with genome-wide association studies. We were able to demonstrate that our results replicate much better. So they reproduce about 9 or 10 times better. And it's because instead of looking for relatively rare variants with high effect sizes, the fact that they're rare means that they tend to suffer from biases in founder populations. And therefore, the results using those particular variants don't translate as well across patients with different ancestries. Ours are looking for combinations of relatively common variants, and those, by their very nature, don't suffer from those biases in quite the same way. So they translate typically an awful lot better across different populations.
Fantastic. And next question, looking at regulatory considerations. We asked, what are the considerations from a regulatory perspective surrounding the use of AI for implementing therapies or diagnostics, such as ones derived from these insights?
Yeah, I mean, this is the topic of a whole webinar in and of itself. But broadly speaking, I think the challenges with AI approaches are the consistency of recommendations that they make is often affected by changes in the training data. So there is a natural tendency to overfit to the training data, and I know a lot of people are working to overcome this, but it remains a particular challenge, and that means that the results don't translate as well as you might expect them to. But that lack of consistency, the changing of recommendations as you add new data in, is a real problem from the regulation perspective. It's really hard for a regulator to come in and say, yes, you've got a nicely deterministic process. It is based on things that clinicians will understand or regulators will understand, i.e. tied back to a particular mechanism. That mechanism can be correlated with a clinical endpoint. That's what you really want to get to. But many of the sort of more black box AI tools don't enable you to do that, and it really does present a challenge in terms of the clinical adoptability and the regulation of those tools. And it's the reason why the FDA has been so active in the context of looking at AI black box models as a software as a medical device classification in the context of LDTs and other tests.
And I think we've probably got time for one last question, and here's a good one to end on. How does industry engage with you, and what partnerships or collaborations are you seeking to translate these findings into clinical applications?
Oh, that's a very good one. So there's a variety of different answers. I mean, obviously this covers a broad range of applications. I think precision medicine has had a lot of impact in oncology and rare disease. As I said, there are many diseases where these approaches could be usefully applied, and it goes everywhere from novel target discovery, through clinical trial design, through label expansion in the biopharma space, so finding other indications in which existing therapies are likely to be effective, out into the clinical validation of new mechanistic and diagnostic tools, implementing that type of differential triage to impact the healthcare economic outcomes, and then starting to think about validating the active protective biology that we identified. Um, I showed 50 odd diseases. We obviously are not able ourselves to progress all of those, and we view partnership as an absolutely fundamental way of taking forward the clinical validation of the benefits of these tools, these approaches, these targets and assets, and the biomarkers associated with them. And anyone who's got access to data, to patient populations, or programs that would benefit from these type of biomarkers, we'd love to hear from them.
Thank you, Steve, and thank you for everyone who's sent us questions. For anyone whose questions we haven't answered yet, I'm sorry that we haven't had time to, but we will make sure that we get back to you via your contact email with a response. And if you can think of anything else that you would like to ask, or any questions you'd like to send through after the webinar, feel free to contact us at hello@precisionlife.com. And that will reach us and we'll get back to you. You can also visit, of course, our website and follow Precision Life on LinkedIn for future updates or details on future webinars, upcoming events, and potential roadshows in and around your area as well. Steve, it's been a real pleasure hearing you present, and thank you so much for giving up your time.
Thank you for setting this up, Noah.
You're very welcome. And thank you all for joining us too. Until next time, goodbye.